Welcome to Food Safety 5, twice monthly reports from the Food Safety Magazine editorial desk to keep you abreast of top news stories and current events impacting the food safety sector. This episode discusses 3 scientific studies on Listeria biofilms and an important E. coli strain with relevance to the leafy green sector and beyond, as well as regulatory updates from around the globe. First, in a new study, researchers from the University of Southampton evaluated the antimicrobial efficacy of chlorine and peracetic acid, or PAA, against Listeria monocytogenes biofilms. The researchers experimented with strains, sanitizer applications, and temperatures relevant to the leafy greens industry. Worst-case scenarios were modeled by applying sanitizers without prior cleaning, simulating neglected niches within a facility. The researchers found that biofilms grew significantly denser at 20 degrees Celsius in comparison to 4 degrees Celsius, underscoring the importance of maintaining cold chain integrity. High concentrations of chlorine, up to 300 parts per million, and PAA, up to 500 parts per million, were ineffective against mature biofilms. The sanitizers were effective against planktonic cells, however, providing evidence of reduced susceptibility of biofilms to chlorine and PAA. Moreover, imaging showed that L. monocytogenes preferentially adhered to cracks and troughs in stainless steel, which complicates cleaning efforts and enables long-term persistence. Although single-agent sanitization proved ineffective in the study, prior research has demonstrated that combination treatments, such as PAA with saturated steam, may reduce biofilm viability. Overall, the findings emphasize that effective control of Listeria biofilms requires integrated cleaning strategies combining mechanical disruption and chemical treatment, tailored to surface material, temperature conditions, and biofilm maturity. Next, a first-of-its-kind study has provided new insights into the genetic and phenotypic adaptations that enable L. monocytogenes to form robust biofilms. The study, conducted by Wageningen University researchers, marks the first application of experimental evolution to study biofilm enhancement in L. monocytogenes. The researchers isolated evolved variant strains, or strains that have developed a specific advantage through evolution, by subjecting 2 Listeria monocytogene strains to repeated cycles of surface colonization, biofilm formation, dispersal, and reattachment. The resulting evolved strains demonstrated up to a 7-fold increase in biofilm production compared to their ancestral counterparts, which were a reference strain and a hypermutator food isolate. Analysis revealed that increased cell surface hydrophobicity was a dominant trait among evolved isolates, correlating with stronger attachment to hydrophobic surfaces such as polystyrene and stainless steel. Further genomic analysis pinpointed specific mutations associated with the elevated expression of one protein, which in turn influenced cell surface properties and biofilm capacity. However, the researchers also found that increased hydrophobicity and biofilm formation was linked to the specific genetic changes associated with the elevated expression of the protein rather than the protein alone, and that the impact of the protein on biofilm formation is linked to its overexpression rather than just its presence. In another ongoing study relevant to the leafy greens sector, a Michigan State University researcher funded by the Center for Produce Safety is investigating a reoccurring, emerging, and persisting, or REP, E. coli 0157H7 strain, investigating the genetic fitness, traits, and environmental conditions that may contribute to its persistence, with the goal of developing a tool to help growers respond to this food safety threat. E. coli Rep EXHO2 has been linked to multiple leafy greens associated foodborne illness outbreaks. Despite its persistence, it remains relatively rare in the environment. The researchers are currently conducting baseline sampling in a leafy greens production region, collecting soil, sediment, water, and wildlife fecal samples. Samples would be analyzed for the soil texture, pH, soluble salts, organic matter, and heavy metals. Of particular interest is arsenic tolerance, which may reside in the accessory genome of Rep EXHO2. This trait could be passed between subtypes and may confer broader stress tolerance. The researchers are also sequencing both new and historical isolates to trace Rep EXHO2's evolutionary origins. AI and machine learning tools will be used to identify environmental and crop-associated traits that distinguish Rep EXHO2 from other subtypes. Moving away from the latest food safety research, to round out this episode of Food Safety 5, here are a few bite-sized regulatory updates from around the world. In the U.S., the joint FDA-USDA request for information on a potential federally recognized definition for ultra-processed foods, or UPFs, has closed, with various stakeholders sharing their comments. Meat industry representatives, such as the National Chicken Council and the National Pork Producers Council, suggested that a definition for UPFs that focuses on processing steps of a food rather than nutritive content could place meat products like chicken nuggets in the same category as foods like candy, despite their vastly different nutritional profiles. Generally, the groups were unsupportive of officially defining UPFs. Other groups representing consumer and scientific interests were more in support of defining UPFs, although their opinions on how UPFs should be defined varied. For example, the Center for Science and the Public Interest advocated for a definition that is scientifically linked to health concerns to be the focus of federal policies, while suggesting the use of a term other than UPFs that would more precisely convey harm. In its comments, the Institute of Food Technologists emphasized above all that a definition for UPFs should clearly differentiate between product formulations versus processing and should not further contribute to the stigmatization of processing, which can be important for food safety and quality. In contrast, the Consumer Federation of America suggested that a federal UPF definition should be grounded in a food's level of processing alone, urging FDA and USDA to resist calls to conflate its UPF definition with other factors related to dietary quality or to exclude certain foods. Across the pond, the UK Food Standards Agency has officially updated the Food Law Codes of Practice after 2 years of consultation and revision. Key changes include a more flexible, risk-based approach to prioritizing initial official controls of new food businesses, allowing for greater use of alternative control methods like remote regulatory assessments, broadening the cohort of professionals who can undertake certain activities to support the delivery of official controls, and the introduction of the new food standards delivery model in Wales. Next, in Canada, the Canadian Food Inspection Agency has enacted regulatory amendments aimed at reducing red tape and enhancing economic resilience in the agri-food sector. These changes affect the fresh produce industry, specifically related to labeling, and hatcheries, requiring salmonella testing for U.S. imports to align with Canadian requirements and providing greater traceability code flexibility. Finally, regulatory agencies based in Abu Dhabi, United Arab Emirates, have launched a strategic initiative to develop a regulatory framework for novel foods and processes, such as alternative proteins and precision fermentation. The new system will be aligned with international best practices, reduce the registration period for novel foods by 6 to 9 months, and introduce a science-based risk assessment approach to approvals. For more information on any of the stories mentioned in this video, be sure to check out the Food Safety Magazine coverage linked in the description. Check back twice monthly on Tuesdays for the latest Food Safety 5 episodes, available at food-safety.com or on our YouTube channel.